Hi, good morning, everyone. So my name is Chee and I'm from NextGene Scientific. So first of all, I'd like to thank you all for joining the sharing session today on the topic, What You Missing Matters, Decoding Full-Length RNA Transcript with Oxford Nanopore. RNA sequencing has always been the subject of interest in addition to DNA sequencing. So this is because DNA need to undergo the process of becoming RNA before carrying out its function in the form of proteins. With the advancement of the sequencing technology that's going through from single sequencing to next generation sequencing and currently the third generation sequencing, so it has led to an unprecedented advances in all fields of biology and medicine. It serves as an invaluable tool for the study of human genetics and the pathology associated with diseases. So all these traditional sequencing technology have undoubtedly made comprehensive transcriptome analysis possible and have led to numerous important developments in science. So nevertheless, there are some important limitations in the field that need to be addressed. One of the most common usage of RNA sequencing is to be used for the gene expression studies. The gene expression studies enable the discovery of differential expressed genes, especially when we are comparing two conditions. So for example, the treated sample versus the control sample, or the normal tissue versus the diseased tissues. mRNA will be extracted from these two comparing conditions, and then will convert into cDNA using, for example, oligo-DT. RNA sequencing is then performed to quantify the transcript number of all the genes in the sample. Then we use the bioinformatic analysis to detect genes that detects higher or lower in terms of quantity between these two conditions. And this gene could be the reason contributing to the differences of the sample. So with RNA sequencing, we are able to identify lists of genes that are upregulated and downregulated. So in addition to the common use of RNA-seq for gene expression study, RNA-seq also actually a very valuable tool for the study of isoform. So after the completion of the human genome since 2003, so we know that our genetic codes has been stored in approximately 3 billion bases in haploid terms. So by deciphering these 3 billion bases, we are estimated to be able to identify between 30,000 to 63,000 human genes. However, does it ever occur to you that whether this number of genes is actually enough to code for everything that we needed for life? So in fact, this number could be just a disguise. This is because a single gene could be not only codable for one, but actually for two and even for three different products. The variation in this RNA transcript structure is a key feature of eukaryotic gene regulation, and it has allowed for over 230,000 alternative transcripts to be derived from. So it's estimated that 95% of genes are producing an average of 3 transcripts per gene. This is possible through a mechanism that we call gene splicing. So gene splicing is an important mechanism that regulates isoform expressions in a cell-specific or time-specific manners. Okay, here I would like to show you different categories of splicing and how different splicing event produces different isoform. So on the left of this image that I put here, okay, so we have a DNA sequence with intron. So the intron is highlighted with a line structure, whereas the exon is the color box. So we have the exon 1 in the form of blue box, exon 2 in orange, then exon 3 in the light blue. So when we have the constitutive splicing, normally let's say in this example, all the 4 exons we join together to form, let's say, isoform 1 from top. Okay. Then in another event, might have what we call an exon skipping. So in this example, the second exon, the orange box, is being missed out. So therefore, we have an isoform that only consists of exon 1, 3, 4. And in another type of splicing, we have intron retention. So a small part or the entire exon is kept. So therefore, we have an isoform that not only have exon 1, 2, 3, 4, but also the intron. Okay. So next, we also have like mutually exclusive exons. So we have the skipping of one of the exon. So next, we also have like alternative 5' splice site that retain some of the 5' or different 5' of the exon, and alternative 3' splice site. Okay. So all these different events of splicing will generate many different isoforms, even though the DNA sequence is actually the same. Now that we have a brief idea of how alternative splicing and isoform work, so you could be still like wondering why we need all this information. Why do we need to study isoform? So let me tell you some of the importance of isoform studies. So number one, transcript isoform expression and usage is a prominent source of variation between healthy and diseased tissues in a number of medical conditions, including cancer. Number two, differential isoform expression and allele-specific effect are known to be important in the susceptibility and progression of diseases, again, cancer, as well as individual drug responses. Number three, alternate splicing can have dramatic effects on the proteins produced. For example, protein-protein interactions may be altered, enzyme function inhibited, or location of expression may be changed. So cell surface protein may instead be secreted. So number four, such splice variation may significantly impact disease risk, disease progression, and drugs response. And last but not least, genetic variants with splicing regulatory sequences can also lead to aberrant transcript isoforms that can cause disease. So isoform is not the only reason that we would like to study the entire RNA transcript. RNA sequencing is also instrumental in identifying fusion transcript present in a growing number of disorders. So fusion transcript representing the products of a fusion gene or the splice together of transcript encoded by different genes. Significant in clinical research due to their association with many diseases, including cancer. One of the very famous example here will be the BCR-ABL fusion genes for chronic myelogenous leukemia. So BCR and ABL fusion genes form when pieces of chromosome 9 and chromosome 39 break off and trade places. So you can see in the diagram at the bottom. Okay. So originally BCR is from chromosome 9, chromosome 22, and the ABL is from chromosome 9. So part of it actually break off and fuse together to form a fusion gene. So the changed chromosome with fusion gene is now called a Philadelphia chromosome. This is actually a hallmark for cancer. So after looking through the importance of RNA transcript sequencing, so now I would like to introduce to you some of the challenges of RNA sequencing. So high throughput RNA sequencing approaches often encounter difficulties with the generation of full-length transcript, mostly due to premature termination of reverse transcription. When using an oligo-DT, they could be reflected in an overrepresentation of the 3' transcript end at the expense of complete sequence. So a mitigation of this problem is presented by the reverse transcriptase of template switching method used
Hi, Chee Hong. I think
Yes.
we lost your voice.
Okay, sorry. Can everyone hear me now?
Yes, it's good now. Thank you.
Okay. Okay. So I think I will go rewind backwards a bit. So I will continue talking about the transcript. Okay. So high throughput RNA sequencing approach often encounters difficulties with the generation of full-length transcript. Okay. So this full-length transcript is because of the premature termination of reverse transcriptase. So as shown in the picture on the left here, if we are a common strategy for reverse transcription is using oligo-DT. So when we are using oligo-DT, the cDNA will transfer. However, sometimes we cannot control like how far we are able to reverse transcribe. It might be having an early termination and not the entire full-length RNA molecule. So this will actually generate a problem with the overrepresentation of the 3' end. Therefore, a mitigation to this problem is by using a template switching method or a template switching oligo. So this actually been shown in the picture on your right. So as a characteristic of a reverse transcriptase, it will actually give you an leaving a CCC shown in the picture there. So we have a template switching oligo that actually ended with a triple G that bind to the CCC. So far, we can actually create a 5' end for the newly synthesized cDNA. So now we have the oligo, now we have the cDNA being synthesis with a 3' end as well as the 3' end ended with a template switching oligo. So this will be able to get a more equal amount of the sequencing of the 3' end as well as the 5' end. So however, this actually poses a new challenges. Okay, because normally the length of the RNA transcript is very approximately 1,500 to more than 5,000. And traditionally, we are using a sequencing machine that's capable of only performing short read sequencing. So the size of this short read sequencing can only sequence approximately 500 bases. Okay, so compared to the aforementioned full-length gene sequence of 1,005 and above. Okay. So to solve this problem, fragmentation of individual transcript to smaller size is required. After the sequencing, bioinformatic analysis is then used to stitch all these short reads back together or map them back to their respective genes. So showing on the image on the left here, we have the reference on the top. So we have the ground truth transcript, the gene A and gene B. So when we are doing mapping of the short reads, okay, so in some cases we actually have a sequence that is actually can map to more than one location. So for example, if let's say the sequence belongs to a highly conserved domain, so this highly conserved domain may appear in gene A as well as gene B. So when we're doing short read sequencing, these 500 bases might be shared between the gene A and gene B. Therefore, in the end, we are unable to map the short reads to the site. Okay. So in comparison, if you are doing the long read sequencing, we actually sequence through the entire transcript, and therefore we get the full length sequences. So we have no challenge for this mapping. Another challenge will be the splicing and isoform. Okay. So the process of alternative splicing, whether a precursor mRNA molecule can give to multiple mature mRNA molecule that has different exon content as discussed in the earlier slide. So this allowed many proteins to be coded by a single gene. Okay. So in order to correctly identify this unique mRNA isoform, it's necessary to sequence several consecutive exons due to their length, which is actually beyond the capability of short read sequencing. Okay, so if let's say we look at the picture showing here, so the lowest part is actually showing the long read sequencing sequence from Nanopore. So we actually sequence through the entire transcript. So for example, we have exon 1, 2, 3, 4, 5, 6, 7, up to 9. Okay. So for the first isoform, Nanopore is actually sequenced through the entire 1 to 9 exon. Isoform 2, which is actually missing the exon 3, exon 7, and 9. So we sequence through the entire transcript again. That's why we're able to detect it. And then lastly, if we have an isoform that missing exon 1, 5, 7, and 9, okay, again, we are able to sequence the entire transcript. So in contrast, if we look at the short read sequencing in the middle, because they are actually consists of some short reads, in the end, we are having some hard time trying to differentiate the isoform. So short read sequencing will require computational reexample to identify isoforms. Okay. However, based on a study by this Stager and their group, it reveals that an automated transcript assembly methods fail to identify all constituent exons in over half of the transcript analyzed. So furthermore, of this transcript with all exons identified, again, over half were incorrectly assembled. These complications are further compounded when reads from highly similar transcripts, such as those from the paralogous genes, are under investigation. So again, rare isoform could be also an issue and remains altogether undetected. Therefore, the main message here today is that short read sequencing is actually missing a lot of biological information. So here I'm showing an image adapted from Woden and Volymers. So we are focusing on the gene called R2C2 here. Okay. So on the annotation, the top pane, we are showing the reference sequences. Okay. The middle, we are showing a short read sequencing that only generate reads on the 5' end. So we are missing nearly the entire middle and the 3' end. Okay. So in contrast, when we are using the Nanopore long read sequencing, we should actually show at the bottom pane, okay, we are actually getting the entire transcript. So about 80% of a transcript is missed by short reads. In another example, okay, so we are actually showing what could we be the missing information. And this is actually a well-known example. So short read expression misses the well-known isoform of CD45 that is actually being used to identify immune cell type. Okay. So the CD45 is actually a frequently used marker for immune cell, and it's a well-established that certain cell type use special isoform of CD45. So you might then be well expected to see a difference in expression across different cell type in the middle panel, okay? But you don't. So all you see is actually some homogeneous expression of CD45 because here they are looking at short read expression that is unable to identify the isoform. So you might also say that, okay, CD45 is a surface protein, so I might use the antibody to differentiate the different isoform. Okay, but even antibody, it could be sometimes unable to differentiate between certain type of isoform. Okay. So what we really need is a sequencing readout that can really directly identify the isoform of CD45. So I would like to highlight today that long read sequencing is actually a method of choice for obtaining and analyzing full-length transcript. So it does not involve any assembly, mapping, and isoform issue. So with long read sequencing, the entire fragments will be sequenced regardless of their length. Long Nanopore reads generated complete transcript isoform sequences circumvent issue with assembly. A full-length cDNA can be sequenced as a single fragment, reducing significant multiple locus alignment issue. Their advantage in splice variant identification has been repeatedly demonstrated. So reliable isoform resolution by nanopore technology makes the de novo assembly of complex alternative splice transcriptomes possible. Okay. So here I would like to show you why nanopore is actually a method of choice for long-read sequencing. Okay, so because we are capable of doing very long sequencing reads. Okay, so for example, the minimum is 20 bases up to a million of bases. Okay, so basically we are able to sequence any fragment regardless of their length. So as you can see in the video here, okay, so the DNA fragment that has been prepared, okay, it will be loaded into the flow cell, and we have a sequencing nanopore here, will be sequenced through the entire RNA fragment or cDNA fragment. So here, I would like to bring you all an attention about an important question. So are short reads really enough? Okay. So bear in mind that up to 95% of human multi-exon genes are undergo alternative splicing. So around 15% of human hereditary diseases and cancer are associated with alternative splicing. And development of 1 in 6 cancers is actually driven by gene fusion. Okay. So I would say that there are some valid questions to ask before you are starting your RNA project or even your previous completed experiment. Okay. So what if I could go back and resequence a sample in my freezer to discover more? Could I find that alternative splicing plays a key role in my research? Will adding isoform resolution improve my chance of getting published? And am I be able to provide my core lab users the latest full-length sequencing technology to add isoform information? So as a comparison of long-read and short-read sequencing, nanopore technologies uniquely deliver high output of long reads that span full-length RNA transcript, supporting quantification and complete transcriptome characterization down to the isoform level. Long read provided by nanopore's devices range from 1 to 100,000 kilobases, making the identification of full-length transcript and de novo transcriptome assembly a brief. Differential isoform expression and allele-specific effect are known to be important in the susceptibility to and progression of diseases and individual drug responses. So here I would like to go to the second part of my sharing, which is actually some case study that using for isoform sequencing. Okay, so there will be 3 sections of case study. So the first section is about isoform sequencing. The second part is about the sequencing of direct RNA sequencing of the RNA molecule to determine the poly A tails length. And we will have the single cell section case study as the last one. Okay. So in this work by Bolisetti group, nanopore sequencing and the Drosophila model system were used for a transcriptome analysis of genes famous for having a large number of isoforms. So namely DSCAM1, RDL, MRP, and MHC. So one of the most astounding findings is this work is that they are able to resolve 7,874 DSCAM1 isoforms with an average identity of more than 90% for full-length alignment. This gene is by far the most complicated alternative splice gene known in nature. And even more amazing is that the exon within each cluster actually share 80% of nucleotide identity with each other. And with nanopore sequencing technology, we are able to resolve. Okay. In this second case study by Oikonomopoulos is actually showcasing the power of Nanopore to perform full-length transcript sequencing. The team used the external RNA control consortium, the ERCC RNA spike inmates, and then generate a cDNA library from human HEK cells. Now, comparing the performance of different sequencing machines, the results show that 90.8% of Nanopore reads were of full-length transcripts. This enables Nanopore sequencing to provide accurate quantification of full-length cDNA molecules. In comparison to the work for Oxford Nanopore just now, so Hargreaves and Molly are able to obtain 88% reads representing full-length transcripts by using Nanopore. Okay. But more importantly, this figure is achieved using a protocol without template switching. So this work shows that even without template switching or template switching protocol is actually not a prerequisite for obtaining full-length transcript when you're using Nanopore sequencing. So the third work from Gleanos group, they are actually doing a revisit to the human transcriptome. As we know, human is actually the most well-studied organism because it's related to us, and RNA sequencing has been done numerous times. Okay, but they are still able to discover some new things when they are using Nanopore long-read sequencing. Okay, so this time when they are using long-read sequencing for Nanopore, with the help of the long-read sequencing, they are able to identify 93,718 alternative RNA transcripts, with 77% of this being novel transcript structures. So this shows that with Nanopore long-read sequencing, we are able to provide new insight to some standard well-studied model or organism. So here we have a work from Clark and co-worker, and they are using the long-read sequencing to study the psychiatric risk gene, the CAC-NA1C in human brain. This gene, CAC-NA1C, is a potential therapeutic target for bipolar disorder and schizophrenia. So this gene has a not well understood isoform structure and expression. So this is the reason is because it's actually having a very long length, approximately more than 13,000 kb. There are many exons within this 13,000 kb, more than 50. And this gene also shows a high level of alternative splicing. So in this study, they're using the Nanopore sequencing, the machine called MinION. They are able to identify 90 transcript isoforms of the CAC-NA1C gene. Okay. Of these 90 transcript isoforms, only 7 has been previously reported. And more interestingly, 9 of the top 10 expressed isoforms were novel, previously unknown transcripts. So with that, I will move on to the next case study focusing on direct RNA sequencing. So one of the unique features offered by direct RNA molecule sequencing is poly-A tail sequencing. So Nanopore is able to perform direct RNA sequencing for sequencing of full-length native RNA molecule down to the poly-A tail level. So in eukaryotes, messenger RNA, mRNA, is augmented with a series of adenine bases at the 3' end, known as the poly-A tail. These tails can vary in size, with the largest being over 250 nucleotides in length, and therefore, beyond the typical analysis, is capability of the short-read sequencing technologies. So research suggests that the poly-A tail length is an important factor in post-transcriptional regulation, and further study may provide new insight into the gene expression and disease. Okay. In this particular research paper by Roche and their group, is actually performing sequencing of the full-length transcriptome of C. elegans using direct RNA sequencing. By using the nanopore sequencing, their group are able to identify dynamic poly-A tail length distribution across the development stages of the model organism C. elegans. So in this study, they found a positive correlation between intron retention and poly-A tail length distribution. Okay, so this is suggesting that poly-A tails may be subject to post-translational processing by deethylation once exported to the cytoplasm. So in another study by Workman and their group, they also actually found similar observation. So they also observed that transcript with a retained intron tend to have a longer poly-A tails than spliced transcript. So this result is consistent with previous observation that nuclear transcript with retained intron has longer poly-A tail due to the activity of nuclear poly-A binding protein and poly-A polymerases. So in addition to poly-A tail sequencing, a further benefit of direct RNA sequencing is to perform viral RNA sequencing for the detection of modification. So it allows the detection of epigenetic modification alongside the sequencing data. So modification on viral RNA, either genomic RNA or RNA transcript, have complex effects on the virus life cycle and slower response to viral infection. So in this study by Lu and their co-workers, so using a TOMPO and XPO pipeline, it reveals more intensive modification in subgenomic RNA than in genomic RNA in siRNA RNA. So modified uretines were frequently identified, exhibiting substantive overlapping between vRNA generated in different cell lines. So now we will move on to the last part of today's case study, which is actually using nanopore technology to do full-length sequencing of single-cell and spatial assay. So for those of you who are moving into single-cell and spatial sequencing, so it's now possible to review isoform level expression from both 10x genomics single-cell and 10x Visium spatial libraries. So on the left here, on the left images here, we have a single cell cluster plot, but actually is generated from a full-length isoform reads. So we can get equivalent cell clustering plus all the advantages of isoforms. So on the image on the right, we will see an example of isoform expression from 10x genomics Visium. So we are looking at expression of the 201 isoform of SNAP25 on the left and the 202 isoform on the right. You can clearly see that in the middle, in the midbrain region, the circle one, there is a strong preference for the 201 isoform and almost no expression of the 202 isoform. So this type of isoform level information is most often completely missed in short-read data set, where you instead see homogeneous expression of just the SNAP25 gene. So in another study by Lebrigan, it showed that it's now possible to discover isoform switching behavior that impact, for example, the brain development. So here we are looking at the expression of two isoforms of the CLTA gene. So if you look at the images here, you can see that the 206 isoform is dominantly expressed in the precursor neuronal cell. As this cell mature, they actually stop expressing the 206 isoform. And instead, we observe the increase of the expression of 204 isoform. So in most cases, this remarkable isoform switching event are not even possible to see. So the authors found 76 genes that show this type of differentially expressed isoform between cell types. So with long-read sequencing, we could have a deeper understanding of the immune response with full-length immune transcript as well. Okay. So on this work at Ben-Hur Lee is in collaboration, they are actually performing a collaboration with Oxford Nanopore team using full-length VDJ sequencers to identify the IGHV gene used by the plasmablast cells in the immune response after vaccine exposure. Okay. By using this data, it's possible to pair the heavy and light chain sequencers, and therefore you can see that the upper left of the heavy chain pairing with the full-length reads. In addition, one big advantage they found is that a better antibody isotype identification is being obtained when we're doing long-read sequencing. Okay, so you can see that the image on the left bottom, there is we are able to identify the IgG1 and IgG3 antibodies, okay, and the changes in the hinge region. Okay, so this region is not covered well by short reads, and the author found those isotopes were often misaligned. Okay, so by using full-length reads, they are now getting unambiguous isotype identification. So next, we are looking at the study by another group. Okay, so they are using long-read sequencing to capture full-length fusion transcript in cancer. And they're able to do so even in the rare cell. So fusion transcript identified on single cell level in acute lymphoblastic leukemia with long nanopore reads. So this group Chen, they identify higher ratio of fusion in leukemia-associated gene, the KMT2A, in the cancer cell versus the healthy control. Okay. So KMT2A fusion confirmed in rare cell population. So another group, they are using the long-read sequencing to discover subtle cellular differences with greater resolution. So they're using the model plant, the arabidopsis. Okay. So in their study, they found that high-resolution cell subtyping are enabled by nanopore sequencing isoform information. So intron retention differentiates cell type cluster as well. So I will end my sharing today with a summary. Okay. So when you're using a short-read sequencing method, you actually only seeing a short snippet of the transcript at either the 3' end or 5' end, and you actually miss the important part of mRNA, including most of the coding sequence. So you could be missing the alternative splicing, the isoform switching, or transcript targeted by nonsense medicated decay. For single-cell sequencing, you might be missing the discovery of comprehensive readout screening of CRISPR, multi-omic, genomic, and expression, complete TCR-BCR sequences, and the isotype of the antibody. So for fusion gene identification, you could be missing the disease target, and the targeted full-length gene panel, and the isoform. So with that, I would like to thank all of you for your attention. And now I actually open the floor to questions.